Okay, so welcome to today's talk, which is about the modeling of neurodegeneration. My name is Stefanovski. I'm both a researcher and a clinician working in the modeling of neurodegeneration and also in the clinics of neurodegeneration. And yeah, I wrote my doctoral thesis in the brain simulation section of the Charité and the Berlin Institute of Health. And I will now give you an overview of what we are doing, but also what is important, in my opinion, to know if you want to apply computational neuroscience and modeling to the complex field of neurodegeneration. So it might be a broad talk, but I hope it will be not too much. In the beginning, I start with some short recap, which is probably is redundant to the talks that Petra and Micha gave the weeks before. But I just want to shortly remind you how we build virtual brain models. So the virtual brain is a software, actually a neuroinformatic platform that is based on two major columns. The first one is the structural connectivity, which you see here, which we infer from diffusion tensor imaging or diffusion weighted imaging. So we extract a stratigraphy, which is kind of the skeleton of the simulation, and then the nodes between the structural connectivity, the brain regions, are the things that we bring to life with so-called neural mass models. So with a technique from nonlinear dynamics. So these are mathematical models that are able to produce oscillations and other neural behaviors. And this gives us the possibility to implement and integrate many different kind of empirical data, either as we directly integrate it to launch the simulation as a structural connectivity, or as I will show in the talk today, for example, PET. But you can also implement other functional data as EEG, MEG, or other, actually all electrophysiological data, because we can project the raw neural activity that is simulated to electrophysiological recordings and compare this with empirical data. So the very first validations have been performed. For example, this early work by Petra Ritter showed that functional connectivity can be reproduced quite easily, and also more complex validations have been outperformed, for example, by Michael Schirner. In this paper, for example, he showed that different empirical phenomena of various scales can be reproduced in the brain simulation with the virtual brain. So, for example, here the opposite behavior of excitatory and inhibitory neurons in different populations could be reproduced in the simulation, very similar to the findings in empirical data. And, for example, also here another particular aspect, the inverse relationship, or the negative correlation between alpha power and inhibitory activity, which is also well known from human empirical data and could also be reproduced directly in the simulation. Okay, so what we are also now doing, and this is also touching the last week's talk, is that we can do these simulations as they are computationally quite effortful, not only on local machine, but also on cloud-based computing, so on supercomputers and clusters. And we are also working together with eBrains, data platform of the Human Brain Project, a European platform, to allow high-performance computing and brain simulation in a GDPR-compliant infrastructure. So in an infrastructure that allows for the compliance with strict data protection laws and high security levels for protection of the patients and the human data protection rights. So As I will now talk about the clinical application, which is neurodegeneration, I shortly want to mention that there are some clinical applications already out there, for example, for stroke, as you see here. So there were several papers that showed that the fitting of a brain simulation to patients with stroke can lead to parameters that can predict the motorical long-term outcome of these patients, like a mathematical microscope. There was also one quite progressed application with epilepsy. So we were able to show, our colleagues in Marseille focusing on this, that you can model epileptic seizures, and this can help to find the epileptogenic zone, which can then help to plan surgery better. So epileptic surgery is sometimes applied for treatment-resistant epilepsy, that you just cut out a particular area, and of course you don't want to cut out more than necessary, and TVB helps to find this area, and there's also so some pilot data showed that this improves the result of the surgery, and there's also a clinical trial ongoing evaluating this. And another single case application was, for example, for brain tumors, where we could also show that the parameters surrounding a brain tumor, where cytotoxic edema takes place, have different characteristics in the simulation than the healthy tissue. So what about neurodegeneration? You might recognize this figure. A similar figure was shown by Petra in the first talk. And for neurodegeneration, we also have these three axes that we can work with. First one is diagnostics. So I will show you how we use simulated data of Alzheimer's disease patients to improve the diagnostic classification between Alzheimer's, healthy controls, and mild cognitive impairment. But we can also always treat the virtual patients. So virtual therapy, there's one application that will be shown in more detail in another talk, which is deep brain stimulation. And in this talk, I will show you how we can treat patients with virtual drugs. And the other aspect, which is also of high relevance, but was also shown in the former talks, is that we can explore mechanisms. For example, here you see a model of healthy controls showing bistability, and so the switching between two behaviors in electroencephalography. And when you look into the bifurcation diagrams and in the underlying the mathematics, you can directly describe how these processes evolve, and therefore you can gain a deeper understanding of what is going on just by reproducing phenomena in a simulation. So it's not only reproducing something to have it reproduced, but then you know the laws and the mathematics behind it that were needed to reproduce it. So one important aspect, if we have a look to neurodegeneration, is that brain simulation takes place on multiple scales. So we have the large scale, which you are probably all familiar with, that is mainly represented by the structural connectivity that we inject to the simulation derived from diffusion imaging, so the structural connectivity matrix. But then we also have other scales. So there is a so-called mesoscale, which is something in between that contains local connectivity kernels, but also in somehow the neural mass models itself. And then we also have the microscale, which is represented by particular parameters of the neural mass models. So, for example, depending on the model, the particular parameters stand for specific receptors or specific biological surrogates as conductivities or so. And these have, of course, biological surrogates on a molecular scale and can therefore be linked to those. So with this together, we can link both macro and micro effects into the simulation. And as an example that I will go more in detail to, we did this, for example, with the Jansen Rib Model, where we have 3 different cellular populations, with the pyramidal cells and excitatory and inhibitory interneurons. And why is it so important for neurodegeneration? Because neurodegeneration takes also place on multiple scales. And even more scales are part there. So first, maybe the common scale is a large-scale connectivity scale. So we know that network effects play a role in neurodegeneration and especially in dementia. So there are controversies about how big these effects are, but I think it's undisputable that changes in functional and structural networks are a crucial part of the trajectory of the disease of a dementia, and in particular of Alzheimer's disease. But you also have, of course, the microscale there. So probably you all know the two hallmark proteins of Alzheimer's disease, which is amyloid beta and the tau protein, and so the phosphorylated tau protein, which are, of course, also on a very microscopical level, and factors like, for example, the misfolding of these proteins and expression of particular genes and enzymes play a role, how these proteins are processed and how they then might lead or might lead not to a neurodegenerative process. And on the same scale, we have, for example, gene expression, but also effects from neurotransmitters, for example, on particular receptors on the synaptic level. And then we also have scales of neurodegeneration that we cannot directly capture in brain simulation, which is, for example, the vasculature, so the damages and macroscopic vessels, but also in the micro vessels of the brain that are important for strokes, but also for clearing of proteins, and therefore also involved in the clearing of pathological and toxic proteins in Alzheimer's disease. And the other important thing is, of course, the immunity, so the role of the innate and humoral immune system, and the role of the microglia, of course, that is an important driver of neurodegeneration, at least in the end phase. As I'm now talking so much about neurodegeneration, we also have to look a bit deeper into what is actually neurodegeneration and what diseases and what phenomena are part of the spectrum. And this mind map gives you an overview how many possible neurodegenerative diseases are there, and it's actually only a subsample because these are only the dementias. And it's not even all dementias. These are just the most common ones. So I will shortly give you an overview what all these diseases are and what this has to do with modeling. I think for the start, we can begin with Alzheimer's disease because it's the most common form of dementia and also one of the biggest health problems in the world. So I will shortly give you an overview about the history of Alzheimer's, its clinics, and the underlying mechanisms. So dementia in general, I think this is an important thing, is not itself a disease, but it's a syndrome. So a syndrome is an assembly of symptoms, and this assembly of symptoms called the dementia syndrome may have various causes. So the most common one is Alzheimer's disease, but it can also be the cause of a lot of different strokes. It's then called vascular dementia, but there are also other causes, as for example the Parkinson's dementia, frontotemporal dementia, or whatever. Also an encephalitis can lead to dementia. So whatever causes the syndrome can be then a dementia syndrome. And this dementia syndrome is consistent of the impairment of higher cognitive domains. Normally it's necessary that almost all domains are affected. And in some definitions, it's also required that at least memory is also one part of the domains that is affected, but there also are some different definitions available. So we have impairment of memory, of speech, of practical motor function, but of course we also find neuropsychiatric symptoms like depression or aggressive behavior, and also more particular neurological functions, as for example visoconstruction. And the crucial point is that what makes a difference from a cognitive syndrome to dementia is that it has to impair activities of daily living. So if it's so severe that the daily living is impaired, then we speak of a dementia. And as I said, that Alzheimer's is the most common one. A short look into the history. So Alois Alzheimer found the first patient in the year 1907. He examined a patient, Auguste Deter, who suffered from severe memory deficits at a quite early age. So she was 56 years old, and this symptom with these severe memory deficits were known in that time, but only for older people. And therefore, he kept track of the case. And unfortunately, some years later, the patient died, and he analyzed then the brain. And in this brain, he found pathological findings that are still parts of the diagnosis of Alzheimer's disease, which are actually the senile plaques and the neuritic tangles. And now we know that in the plaques we have amyloid beta, and in the tangles we have tau protein, which Alois Alzheimer was not aware. And interestingly, the patient Auguste Deter suffered from a rare monogenetic form of Alzheimer's disease, which makes about 1% of the Alzheimer's cases. And this is also the reason why she suffered in such a young age from this disease. Interestingly, therefore, Alzheimer's have not found the sporadic Alzheimer's disease, which is the most common form, but another one. And only years later, people found out that the so-called senile dementia show exactly or very similar brain changes and are therefore also part of the same disease family. Now again, back to this figure. So what is the cause of Alzheimer's disease? And the easy answer is no one really knows. So there are a lot of controversies, especially of the role of the vasculature and the immune system, and of the role of these two hallmark proteins, and also of many other factors. So there are also theories about infections causing this. Maybe you have heard that also some studies show increased risk of Alzheimer's after COVID-19, although this is very preliminary results, but also some findings show a relationship to herpes infections and so on. So we know a lot, but we also don't know the actual cause. But what we know is that these two proteins are at least the biomarkers for the trajectory of the disease. So there are also is a particular temporal and spatial pattern how these proteins evolve. And these are described by Heiko and Eva Brag, two neuroanatomists that first described this in the 90s. And for example, for the amyloid, you see these three stages, beginning in the temporal lobe and the medial temporal lobe, then going into the limbic system, and afterwards spreading over the whole cortex. And even more distinct and more strict is the pattern for tau, which are normally when you refer to Brag stages, or more correct, it would be Brag and Brag stages, because both were involved in this classification. There, typically the start is in the transentorhinal cortex. It's an area surrounding the hippocampus near the parahippocampal gyrus, and then afterwards the medial temporal lobe and hippocampal area, and then afterwards the neocortex. So similar distributions, but not exactly the same. And these the temporal pattern is also typically that we see both amyloid and tau changes long before brain changes appear, and therefore also long before symptoms appear. So first, this is a very simplified figure, but normally we consider that first amyloid and tau biomarkers get positive, and then some years later you can see manifest brain changes, as for example atrophy and MRI. And even afterwards, often you clinically see a decline that shows the difference from the control group from healthy aging. This is also the case because the brain, of course, has a a very good and elaborated capability to compensate damage, and therefore it can be damaged for many years and compensate it without losing function, but then sometimes it decompensates, and then you see sometimes a very fast decline. And important is this because we have, before we can detect the biomarkers, this is something that is not really interesting for research, because these are not patients that are not attributable for early diagnostics. And it gets interesting when the biomarkers cut the threshold so that we can detect the early disease, because this is then the window where we might have the chance to treat the disease if there would be a treatment that slows down the disease, which is actually not the case, to my knowledge. And therefore, this in my opinion, this window between the starting of biomarker detection and the starting of clinics, which first typically goes into the direction of mild cognitive impairment, is the most interesting field, because there probably you can save most of the brain. And afterwards, especially when the manifest dementia appears, a lot of the disease process is already fulfilled, and it is very probable that then the treatment options are very, very reduced. Even if we would have a causal treatment, it would be probably much more effective when we start early. And therefore, this is the main reason why we want to early detect Alzheimer's disease. Although for now it's more academic, but hopefully maybe soon we will have an option to treat it then. And how can we do this detection? This is also a big controversy, because the current definition of Alzheimer's disease is still a definition from 1984, which is simplified exactly this. So there is a definition for probable Alzheimer's disease, which contains just a dementia syndrome, so cognitive impairments that impact activities of daily living. And in addition, 2 cognitive domains has to be affected. Memory has to be part of these domains, no conscious disturbance, then an age range, which does not make so much sense. Probably it was in the 84s. They were not aware that sometimes, in very rare cases, also people can get Alzheimer's when they are younger or older than this age, and the exclusion of other causes. And then there is also a list of further findings that might support this, but they are not part of the definition. And actually, when we look at this definition, it's just, probably just the definition of a dementia syndrome with some probability of being Alzheimer's disease. And this is also countercarried by the lower definition of possible Alzheimer's disease, which is just a dementia syndrome and the exclusion of other causes. So just actually, it doesn't, in my opinion, it's a possible Alzheimer's disease, but it does not say more than Alzheimer's disease is possible because there is a dementia. But this is important to know, because this, in the paper that McCann wrote in 1984, they wrote that this should be used in research studies as a correlate for dementia or severe cognitive deficit. And actually, this is also the case in most of the studies. So in almost all studies, except maybe in the last few years, but even then sometimes when there is spoken about Alzheimer's disease, you have to expect that it's this possible Alzheimer's disease, and actually it's just dementia, and therefore you would expect that about 30 to 40% of these patients do not suffer from Alzheimer's disease, but from something else. And what else could it be then? So here is an overview of a cohort of very old patients, almost 100 years old. And when you look at these patients, about half of them have dementia and a quarter have mild cognitive impairment, while a quarter has not such cognitive impairment. And if you have a look to the dementia patients, you see that it's about 2/3 of Alzheimer's disease, and then in a smaller amount other dementias, which is Lewy body dementia, vascular dementia, and here's the category of other diseases. And if we have then a look back into the definition. How can we make this probable Alzheimer's disease a secure diagnosis? Is actually the same criteria as for probable Alzheimer's disease, and in addition, they require histopathological evidence, which is done by an autopsy or a biopsy. And as we normally do not perform a biopsy because it's an invasive procedure and a surgery to the brain, and it has complications as bleedings, infections, and so on, normally this is only done by the autopsy. So the diagnosis of definite Alzheimer's disease is basically only post-mortem possible, and therefore only very few studies use this definition, as you need a very long ongoing study to, in the end, perform the autopsy and make sure that your diagnosis was true. So looking back to this data, we can have a look, okay, what tells us the autopsy when we have this? These are the clinical diagnoses. And if we then want to know about the other neurodegenerative phenomena, we see at the same cohort that Alzheimer's pathology is almost the same. So 2/3 of clinical diagnosis and about 60% of Alzheimer's pathology in the brain tissue. But we also see, for example, a third of TDP-43 pathology, which is a main protein for frontotemporal dementia and amyotrophic lateral sclerosis, which is not captured at all by the clinical diagnoses. We also have 15% of Lewy bodies, which is also nowadays a known phenomenon in Alzheimer's disease, and no one really knows what is the role of them. And we also see, of course, 75% of cerebrovascular disease, so blood vessel impairment, which of course also contributes to the cognitive performance of these patients. So we see, okay, it's not that easy. Probably we don't have a strict disease, especially in the very old population, that we don't have Alzheimer's disease, which is only one pathomechanism. So it's often a mixed pathology. And this also gets obvious if we have a more detailed look to these tissues. So this was a study where they took only the autoptic secured Alzheimer's disease patients. So where just a light microscopy reveals the diagnosis and fulfills the pathological criteria of Alzheimer's disease. And they then performed immunohistochemistry to look further for other proteins. And for the Alzheimer's patients, we saw that although amyloid and tau were in 100%, because this was also the definition of the diagnosis, we also saw 55% of alpha-synuclein, which we know from Parkinson's disease, and 40% of TDP-43. So we still have other neurodegenerative processes that take place even in the gold standard diagnosed Alzheimer's patients. And interestingly, also these patients without brain changes, so without clinical phenomena and also no atrophy, no signs of neurodegeneration, show about half of them show amyloid plaques, and almost all of them show tau tangles. So this is also important to keep in mind when people trust too much into the early biomarker diagnosis. We have to keep this in mind. We actually don't know if these patients never would develop Alzheimer's disease or if they just have a protective factor that we are not aware of. And therefore, a new diagnosis criteria catalog was established in 2018 by Clifford Jack et al., nuclear physician. And so, And there we have a classification which is called the Atn classification, which takes into account these biomarkers. So A for amyloid, T for tau, and N for neurodegeneration. And if we have a look to those criteria, so A considers either amyloid ratio in CSF or amyloid PET, and tau refers to the phosphorylated tau, which you can measure also with tau PET and also with tau in the CSF. And neurodegeneration, there are many possibilities, so you can get it first with total tau in CSF, with atrophy and MRI, or with signs of hypometabolism in glucose PET. So you see always PET is possible, but always also lumbar puncture is possible. And in addition, we also have the dimension of cognition. So you can also give in this classification letter for the cognitive impairment. And this leads to the fact that you can then build more precise diagnoses. For example, here you see amyloid PET and tau PET are positive, and MRI shows atrophy, so it's A plus, T plus, N plus. Here, for example, we see only amyloid is positive, so it's A plus, T minus, N minus. And here, for example, both are negative, and you still see an atrophy, so it's A minus, T minus, N plus. And in addition, you can have a look to the cognitive performance. So, for example, here it was Alzheimer's disease, so this makes sense. Here, for example, it's someone who is cognitively unimpaired, so it might be preclinical Alzheimer's disease, but it might be also someone who just has amyloid in the brain, and we don't know what happens with this group. And here, for example, it also makes sense that we have another neurodegenerative diseases that independent from amyloid and tau, which was actually a autoptic secured TDP-43 encephalopathy in this case. And therefore, you can imagine we open a spectrum, which makes total sense, in my opinion, that here Alzheimer's disease is actually a spectrum that has different constellations of a biomarker profile and also different constellations of cognition. So we can, for example, say that the classic Alzheimer's disease would be Alzheimer's disease with dementia, so A plus, T plus, N plus, with the clinical syndrome of dementia. But we can also have A plus, T plus, N plus cognitively unimpaired, then it's preclinical Alzheimer's disease or someone who has a protective factor and so on. And this classification is not perfect, but it allows more precise diagnosis, and it will enable more precise and more accurate studies, because then we can actually know for which group, for example, a new drug works or works not. And it's not just dementia where we don't know what is the cause. And this is also part of this idea that, in principle, this classification is also open to other factors. So the classic, for example, the very classic sort of Alzheimer's disease is that you first have amyloid deposition that leads to tau hyperphosphorylation, afterwards leading to neurodegeneration, which leads to cognitive impairment. But there are also other possibilities thinkable. For example, vice versa, the tau is the beginning. But it's also possible that another factor, for example, the immune system, leads to the deposition of both amyloid and tau, which afterwards leads to the degeneration. Or what I've also find very interesting, for example, the idea that an independent factor leads itself to the neurodegeneration, and as a side effect leads to amyloid deposition and tau deposition. This is all in principle capturable with this classification, and therefore this will probably in the future help a lot to understand the disease a bit more. To have a look, just an outlook how well this works. This is, I know, a complex figure, but very interesting. Shortly said, this shows for different preclinical classifications what in the end came out, so if these patients had dementia or not. And for example, you can see here for the A plus, T plus, N plus group, it's quite accurate. So most of these group also has, in the dark way, have had dementia clinically. But for example, for the other groups, it's not completely sure. So for example, only A plus, the only A plus group has a low rate of people with dementia. And the only two groups which are actually or the three groups which actually are quite well in predicting the real appearance of clinical dementia is everything that contains positive tau pathology. So this is maybe one of the first studies which I hope that we will have further in the future that help to understand these groups more in detail. Okay, so this was about Alzheimer's. So I will just very quickly also give you an overview of the other causes of dementia, and then directly switch over to the simulation of mainly of Alzheimer's. So now we come back to this mind map, and I just want to shortly mention that in the spectrum of the amyloid beta related diseases, we have, besides Alzheimer's disease itself, also some variants of it. So first, there's an early onset Alzheimer's disease, which is just defined as Alzheimer's disease starting from an earlier age than 65. And we also have the monogenetical forms, as I mentioned, for example, the first patient of Alois Alzheimer with presenilin 1, presenilin 2, and APP mutations. Then there's also the cerebral amyloid angiopathy, which is also another thing because it leads to microbleedings and so on, but also related to the same amyloid protein. Maybe a bit too much for today, but just important to know that this exists. And there are also 3 classical atypical Alzheimer's disease entities, which is the posterior cortical atrophy, something where problems in visual perception appear. Then also the logopenic variant of the primary progressive aphasia, so where speech is in the beginning of the disease, and also a frontal variant of Alzheimer's disease where frontal symptoms as disinhibition or aggressive behavior might appear first. So this is just, they're all rare, but can appear, and it's important to know that not all cases of Alzheimer's disease start with memory deficits. Then we also have the tauopathies. So tau itself can also lead to neurodegeneration. So, and besides Alzheimer's disease, I think the most important ones are 2 atypical Parkinson syndromes, which is the corticobasal degeneration, where you have strictly one-sided Parkinsonism, and the dementia and the so-called alien limb phenomenon, that one limb seems to be not part of the body. And the other tauopathy is a PSP, progressive supranuclear palsy, where you have axial Parkinsonism and particular eye movement disorder, which is very characteristic. And interestingly, also some other things lead to tauopathy. For example, the chronic traumatic encephalopathy, which is very interesting because this is maybe you know it as the so-called boxer's dementia. So this often happens in sports where you have a lot of repetitive head traumata. So, for example, in boxing or in American football, and sometimes years after this exposition, a tauopathy starts and leads also to Parkinsonism and hallucinations and neuropsychiatric symptoms. And this is interesting because there's one theory that just the axonal damage from these repetitive traumas lead to more tau protein, which is part of the microtubules, so actually part of the axons in the brain. And when you have too much tau, then probably it starts to get phosphorylated, and once there is tau deposited in the brain, it has a behavior like the prion protein and starts to distribute along the axons and infects the brain, actually, and therefore you can induce a tauopathy. And another rare condition where tauopathy is induced is the so-called Eglon 5 encephalitis, a very rare autoimmune encephalitis where in the beginning you have acute encephalitis, but afterwards this leads also to a tauopathic degeneration of the brain. And this is very interesting because it shows that really tauopathy is something that can be induced. Yeah, and then also maybe, but this makes it more complicated, that also half of the frontotemporal dementias are also caused by tau. Yep, and the other frontotemporal dementias are caused by TDP-43, which is a protein that is, so it's a TAR DNA binding protein of the size of 43 kilodalton. And it's most well known for its role in amyotrophic lateral sclerosis, where it plays a role, and therefore also in the overlap form, which is the frontotemporal dementia with amyotrophic lateral sclerosis. So both diseases have an overlap of about 10 to 15%. And there are also other variants of frontotemporal dementia that can be caused by this. So it's a classic behavioral form with aggressive behavior, disinhibition, and so on. And then there are two forms of the primary progressive aphasia. So yeah, it's a primary aphasia, the semantic variant with loss of lexical knowledge, actually, and the non-fluent variant with agrammatic speech and problems in the syntax of the speech. Yes, and then also there's also another rare condition, which is the right temporal frontotemporal dementia, which is actually the mirror disease for the PPA, which is shown by depressive syndrome and psychomotor slowing, and interestingly, prosopagnosia, so the impairment in the recognition of faces. And also interesting, because it was just first described a few years ago, is late. It's the limbic predominant age-related TDP-43 encephalopathy, which is similar to the hippocampal sclerosis, and actually shows a very similar phenotype to Alzheimer's disease in very late age, and was identified to be caused by TDP-43. And yeah, for decades we have subsumed these patients as Alzheimer's disease patients. And yeah, you can also imagine those patients were also part of clinical studies with Alzheimer's disease patients. And this is probably also part of the problem that drugs don't work there, because of course we don't find a drug that works against all neurodegenerative diseases. We need different drugs for different diseases and different subgroups. Okay, so the almost last part are the synoclinopathies. So it's these are diseases where alpha-synuclein gets together to Lewy bodies, as for example in Parkinson's disease, and later on often in Parkinson's dementia, but also into atypical Parkinson's diseases, which is the Lewy body dementia with hallucinations and Parkinsonism, and the multiple systems atrophy from the Parkinsonian type and the cerebral type, which includes vegetative symptoms. And yeah, this will also be part of a later talk in the summer school, talking about deep brain stimulation, because although not a causal treatment, deep brain stimulation is a very effective symptomatic treatment for Parkinsonism. Yes, and later, so much rarer than the other cases, but also well known because of the BSE crisis in the late 90s, are the prion diseases. So just to mention them, Creutzfeldt-Jakob's disease is the most common one, which leads to a very aggressive, very rapidly progressing dementia, also very fastly involving all other neurological functions, and unfortunately leads to death in often only a few months. And there are even more rare diseases that are hereditary, which is the fatal familial insomnia, and the Gerstmann-Straussler-Scheinker syndrome, but I think this is probably too specific. Yes, and the last point for the clinical aspects here is that there are also treatable causes of dementia. I think this is important to know, because this is also, in my opinion, one major reason why we have to make good diagnostics for dementia. Because, of course, you can say if a 75-year-old patient come to you and has memory deficits and so on, and you can say, okay, why should I do diagnostics? Because there is no treatment. Probably it's Alzheimer's disease. It might also be something else, but there's also no treatment. No, because there are some rare conditions where you can treat patients. And I think the most important one is a major depression, which is, of course, also a severe disease, but it's better treatable than neurodegeneration. And then also some more rare conditions, for example, infections or vitamin deficits and so on. Important because getting diagnosed more common and more common is autoimmune encephalitis. I think most important is LGI1 encephalitis, which can show a phenotype extremely similar to Alzheimer's disease. For the more acute forms, for example, the CNS vasculitis, and yeah, the other things are, I think, more rare, so maybe only for clinicians of interest. But I think it's important to know that these exist, and you have to keep this in mind when you do diagnosis and also when you work on biomarkers, because positive biomarkers for Alzheimer's help you also to exclude other causes. Okay, so now I'm through with the clinical and background part, and I will directly go to the modeling part. And as I actually almost have the half of the time, I think now would be a good time for the short break. Okay, so I would now continue. So now I speak about how we can model this in the virtual brain and how we modeled this already. So for Alzheimer's disease modeling, we first thought of all the stuff that I just explained, and we focused on one aspect, which is the amyloid pathology, which I already explained. So we have the position of amyloid plaques in the brain, and this leads to several effects there. So independent of the question if this is an effect of neurodegeneration or itself leads to the neurodegeneration, we know a lot from in vitro studies about what effects has amyloid beta 2 neurons. And one of these effects, which is very prominent, is disinhibition, that actually it impairs inhibitory neurons in particular. And as these inhibitory neurons are affected in their function, this leads to disinhibition of excitatory neurons and therefore to a hyperexcitation. And this is very interesting because it fits together with a lot of other findings in Alzheimer's disease. So, for example, in Alzheimer's patients, we have increased epileptogenicity, and we also have effects that are called excitotoxicity. So, too much excitation can lead also by calcium-mediated signaling cascades and so on to more cellular death, and therefore also directly to neurodegeneration. And to model this, we took a generic structural connectivity matrix. So we used data from the Alzheimer's Disease Neuroimaging Initiative and had their patients with Alzheimer's disease, mild cognitive impairment, and healthy controls. And we took the generic connectome from the healthy controls just because we wanted to have a healthy connectome and then wanted to infect it with the individual amyloid beta distribution, introducing this hyperexcitation and disinhibition. And that looked like this. So it's a typical structural connectome. When you draw it as a graph, you see very nicely that the hemispheres are separated, the brainstem is in the middle, and the hubs are subcortical regions which have a lot of connections. So when we have a more detailed look how we can implement this disinhibition phenomena into the brain network model. And therefore, when we look into the literature, we see mainly two direct effects of amyloid beta 2 inhibitory interneurons. First is a decrease of the amplitude of postsynaptic potentials, and the second one is a decrease of the firing rate of inhibitory postsynaptic potentials. And when we then had the, this is also together, sorry, the order is not correct. This is what I mentioned before, that this fits well together to the phenomena of increased epileptogenicity in Alzheimer's disease. Here you can see EEG recordings via foramen ovale electrodes in Alzheimer's disease patients, so invasive recordings that show epileptic or epileptiform activity in the hippocampus in Alzheimer's disease patients, which are not appearing in the surface EEG, and which are also part of this hyperexcitation. And we decided then to implement this directly in the transmission from the inhibitory interneurons to the pyramidal cells in the Jansen Rib Model. This is the model that I've shown earlier, which has these three biologically quite plausible cellular populations. So pyramidal cells and inhibitory and excitatory interneurons. And in this connection, we wanted to implement the effect of our beta, and actually wanted to implement that the postsynaptic potentials we slowed down the firing rate so that more amyloid deposition leads to less inhibition. And this is also interesting because we have not modeled this, but from former modeling approaches, we also know that that this hyperexcitation can also directly infer with memory consolidation, because this is a phenomenon described also already in the 90s called runaway synaptic modification, which just expresses that when you have too much excitation and too strong plasticity, you cannot consolidate memory quite well because of interference reaction. So you always need an inhibitory factor that slows down the activity a bit so that useful memory consolidation can take place. And this was something we also wanted to be part of this model. And in the end, we came up with a quite simple sigmoid model. So here you see the effect of amyloid beta from PET on the x-axis, and on the y-axis you see the one parameter of the transmission from inhibitory interneurons to pyramidal cells, which is the tau e, which is the inhibitory time constant, as we wanted to slow down the firing rate. And the equations for this are these, which look now maybe quite complicated, but actually it's a standard sigmoid. So you can, you have different possibilities to describe a sigmoid. So we here, it shows a exponential form. You can, of course, also use a trigonometrical form, but we saw that's much easier to use exponential equations than trigonometric ones. And yeah, then we just define steepness and so on, and then you get exactly this curve. And the cutoff values were inferred from the literature, so that we know that, for example, at about 1.4 of the standardized uptake value ratio in PET, regions are considered as positive with amyloid. Therefore, we took this area, and the upper border, we took the 95th percentile of the cohort, so that we have a sigmoidal decay inside of this area. And when we then look at the effect, what does this with the postsynaptic potential, we see exactly this slowing down the delay of this potential. And what were the effects? Interestingly, the effects were exactly what we expected, which was slowing in frequencies, and in particular a switch from alpha oscillations to theta and delta oscillations. And this was something that only happened in the Alzheimer's disease group. So you can see here that especially this peak at about 4 Hertz, which is on the border between theta and delta, in EEG and also in local field potentials, appeared in the Alzheimer's group, which did not take place in the healthy controls in the MCI group. And actually, important to know, this is simulated electrophysiological data. So the only heterogeneous factor was the amyloid PET that was injected. So just because of the hyperexcitation, this led to slowing. And interestingly, it was not just a slowing, so we just not switched from alpha activity to theta activity, but the findings were a bit more complex, which is also part of the game in brain simulation, because of course you are always doing this in a big parameter space, and the behavior depends on each of these parameters. And therefore we had a look to the behavior across the global coupling parameter. So this is just the scaling factor that scales the effect of large-scale connections. And inside of this range, we saw very different behaviors. So, for example, here in this Alzheimer's disease patient, we saw that brain regions had both frequencies. So we had some bimodalities, so alpha regions oscillating in alpha oscillations, and also regions oscillating in theta oscillations. And afterwards, the alpha oscillations get slower or get less, and the brain synchronizes in the slower regime. And this was a very typical behavior across the global coupling factor G for the Alzheimer's disease group. We see, for example, here another thing where you also see some harmonic structures, and this did not happen much in the healthy controls. In the healthy controls, we actually mostly saw another phenomenon, where in areas of low global coupling, we saw alpha activity, as you would expect it in physiological cohort. And afterwards, we saw no activity at all, which is of course not a physiological phenomenon. This was an effect of too much coupling in the model. But interestingly, although this is not plausible, this is maybe nothing we wanted to simulate, it's still interesting to differentiate between the groups, because even with bare eye, of course, we can easily differentiate this pattern and that pattern. So therefore, this was something we later on also used for classification with machine learning, which I will show later on. Therefore, just important to have a look here that also the phenomena in brain stimulation outside of the physiological range are sometimes of interest, because it's a simulation. You can bring the brain to every state you want to have it in, and therefore you can also explore what it does in, for example, an overcoupled regime, and this can still be interesting, for example, for diagnostic classification. And this slowing was interestingly also only, so it was of course, as we would expect it, depending on the A-beta load. So with higher load of A-beta, we had lower frequencies, but this relationship was only there in the Alzheimer's disease groups. So of course, in the healthy controls and the MCI group, we also had regions with more and less A-beta, but there we don't saw this relationship. We only saw it in the Alzheimer's disease group. And when we then had a closer look of why this happened, of course, the reason were the distribution of amyloid beta inside of the brain. So here we see two participants where we actually also shuffled the distribution. So we found also out that the actual regional distribution is not so important in this model as the numerical distribution of some regions that are strongly affected with amyloid beta and other regions that are not so strongly affected. And for example, here you see that this Alzheimer's disease patient has this strong cluster of regions oscillating in the slow regime. And we have a look into the histogram of amyloid beta loads. We see that the patient has a lot of regions with a high load. And we classified them already into the alpha regime, the theta regime, and the bimodal regime, which I will show you on the next slides then, based on bifurcation diagrams that we derived from uncoupled systems, which you can use to estimate the behavior of these nodes then also in a coupled system. And it worked quite well because, for example, here you see that this patient shows, especially for lower coupling areas, much more alpha activity, and consequently you also see in the A-beta distribution more regions that lead to this alpha oscillations. And if we have then a look to these bifurcation diagrams, we can see why this happens. So in this alpha regime, so with low values of tau e, we see that there's only one limit cycle, which produces actually alpha oscillations. So depending on the input, which is, of course, a function both of the large-scale connectivity, but also of global coupling, we get to one of these places on the bifurcation diagram. And here there are only two possibilities, which is a stable focus, which in the absence of noise does not lead to oscillations, and the alpha limit cycle, which leads to alpha oscillations. And therefore, we see in patients with a lot of regions in this alpha regime, we see a dominant alpha rhythm because this limit cycle produces alpha activity that is spread along the brain. And then there's an interesting area where we have two limit cycles. So this is only a very narrow range of tau e, where you still see here this narrow alpha limit cycle, and then a second limit cycle appears, which produces slower theta to delta activities. And afterwards, just increasing it a bit more, we get to then a regime where the alpha limit cycle fully disappeared, and we only see the slow limit cycle, and therefore those regions lead to presence of slow oscillations. And then you can imagine we have in this case, we had 379 brain regions that all have their own burden of amyloid beta, and they are all in their own dynamical condition. And what does the simulation is that these are interacting, and this then leads to other interesting phenomena. So here, maybe just you see the dependency of the tau e and global scaling factor, but it's in principle similar to what I've shown you before. The more interesting part is this, because then we wanted, of course, to know, okay, what are the network effects? Because what I've shown you before was not so new, because then we would not need to couple the system. But it was good that we coupled it, because we found out that the slowing was centralized. So the A-beta distribution was not. So if you look here, for example, so the A-beta distribution was not strongest in the hubs of the network. It's actually quite equally distributed, also in peripheral regions. But the theta rhythm was quite strictly focusing to regions with high degree, so with a lot of connections, which you also see in the scatter plot. There was a almost linear dependency between the degree of a region and the probability of showing the theta rhythm. And this even was stable when we shuffled the A-beta distribution. So even with random distributions of A-beta, the slowing always focused to the central hubs of the network. And this is very interesting, because we saw the same phenomenon for the hyperexcitation. So here we just look to the relative firing rates, so relative to the baseline firing rate of the brain. And without A-beta, there is not much heterogeneity in this model. And then you see when we introduce amyloid, we get heterogeneity, but for healthy controls and MCI patients, we see, okay, most of these regions still are at the baseline, so not much hyperactivity and hypoactivity taking place. And for Alzheimer's disease, we saw both. We saw some regions going to much higher activity and some regions going to much lower activity, which of course makes sense, because hyperactivity also leads to hypoactivity in somewhere else when we use a relative measure, of course. And when we had a look where this hyperactivity takes place, we saw again this pattern. So the hyperactivity was centralized to the hubs of the network, again quite strictly, which also in this case makes sense, because of course hubs have more connections and therefore can also receive more activity input, and therefore hyperactivation also mainly gets focused to these hubs. And this is very interesting, because we were able to see that, so two phenomena that we wanted to link, which is hyperexcitation and slowing, are not produced in the hubs of the network, because they can be produced even in the periphery and even in the random distribution, but the effects then hit to the central parts of the network. And this is interesting, because it could explain mechanistically how, for example, change in excitability caused by amyloid beta in the periphery of the brain can lead to a severe dysfunction of cognition, because it does not have to affect the central parts of the network itself. It can just corrupt the network, and therefore also then leads to slowing and hyperexcitation and excitotoxic degeneration also in these central parts. This is what we have described in this picture. Okay, and as I said, we also were interested in virtual therapy. So as I've shown you that we introduced a model of hyperexcitation, it would make a lot of sense to do something against this hyperexcitation. And fortunately, one of the drugs that are available for symptomatic treatment of Alzheimer's disease is memantine, which actually does exactly that. It's an NMDA antagonist. So it reduces the NMDA-ergic transmission, which is the main receptor of the glutamatergic transmission. And we just wanted to figure out if we can reverse these effects when we apply virtual memantine, and how we applied virtual memantine. So while A-beta affected the connection from inhibitory interneurons to pyramidal cells via the time constant, we just reduced the scaling factor of the excitatory interneurons to the pyramidal cells by memantine, and we tried out different amounts, and it worked best with a reduction of 25%. So we reduced the NMDA-ergic transmission a bit. So you can see this here, that is C31 is the parameter of NMDA, and this leads then to a sharper sigmoid here, depending on the input. And the effects were very interesting because the slowing of Alzheimer's disease patients that we have seen before, especially in this physiological area of low coupling, was reversed. So in this line, you see the slowing without memantine, and then by reducing from another side the excitation, this led to normalization, and the Alzheimer's patient showed then the same rhythms as without the drug. And interestingly, this is also described as a clinical effect of memantine. So when you record an EEG, in Alzheimer's patients, you often see these theta slowings, and after memantine, you get an improvement and you get again more alpha activity. So just from the theoretical background and the mathematics, we could reproduce a phenomenon that was observed in empirical science before. And also, not surprising, the effect of the memantine was most strongly in the hubs, where also the most excitation took place. So although it was applied homogeneously to the brain, it acted mostly there where also the activity took place. Okay, so this was a therapy application, and I now also want to explain how we were able to improve diagnostics by this model. And this is the second study with actually the same data, where we used machine learning, and we compared two approaches of machine learning. The first one just took into account empirical data, so it was actually amyloid PET, tau PET, and volumes in MRI. And then we have had a second data set where we just use the simulated local field potentials from this excitability model. And then a third option was then both together to compare if we could, just with this simulation that is of course based on the amyloid PET, we could improve the classification of Alzheimer's disease, because maybe our idea was that it provided additional information that was hidden in the amyloid PET. And interestingly, when we look into these features, you can see it here. So in 3 different colors, the different disease groups, Alzheimer's, healthy controls, and MCIs. And as you can see, of course, they are different. So here the dimensions are local tau PET values and A-beta values and the frequency. And the patterns are, of course, different, but you also see that these clouds are overlapping. So it's not eligible for a classic biomarker where you just cut a line between those things and say, okay, everything above this line is Alzheimer's. This is here not possible, but for machine learning, this makes sense. And we also saw then here, for example, these things that especially the MCI group show in the local field potentials a different pattern that is not available in amyloid and tau measures itself. And we used quite a complicated, not so complicated, but a bit multiply cross-validated approach for the machine learning, just because we only had 33 subjects, which is for machine learning a small cohort. And therefore, we also used classic or traditional machine learning classifiers, which was actually for the classification a support vector machine, and for the feature selection random forest. So 2 classifiers put them together, and overall we then had 73 million classifications. And what came out, we really found out that the simulated and empirical features are equivalent, so the simulated even performed a little bit better, but there was not significantly different. So the simulated features were as good to diagnose 3 groups of healthy controls, Alzheimer's and MCI, as the empirical data. But both together were then even better. So we improved the classification of about 0.1 of accuracy, or in this case, the weighted F1 score. So which is for a 3-class problem already quite a lot. And even more interesting was that the features that we that had the highest importance. So we took the feature importance from the random forest feature selection, and interestingly, these features made total sense, which is of course which is not always the case in machine learning. You all know maybe the problem of the black box of machine learning. So it's always good to have a look if the features that were selected make sense. And here this was the case, because from the frequency we saw, for example, typically occipital frequencies and thalamus frequencies, so which are part of the rhythm generator, and also the in the occipital lobe where typically the alpha rhythm takes place, and also the A-beta and tau features made sense. So it was, for example, entorhinal tau, when you remember, which is Bragg stage 1 and near the hippocampus, and mesiotemporal amyloid beta in the inner part of the temporal lobe. So also from the biological perspective, the features that were selected by this approach made total sense. So you can see them also, and it also the distribution of feature importance was plausible. And this was very nice because it actually showed us not only a possibility to improve the diagnostics, but also showed us how we can validate that this is really based on the information that is part of the feature space. Okay, so now we have finished with that, and the last thing that I want to speak of is how we can now further integrate biological knowledge into this neurodegeneration modeling. And the idea behind this is that what I've shown you before is just a model. So it was the first model and the pilot study, but the idea that I initially presented, that we take the disinhibition, was of course an arbitrary choice. So there are of course also many other phenomena in Alzheimer's disease that are also maybe applicable for brain simulation. And often, of course, in the end, researcher has to do normally not an arbitrary choice, but maybe an educated guess or a very well justifiable decision. But it would be, of course, better if we would have a more systematic approach, and if we could just with algorithmic means extract the knowledge about Alzheimer's disease directly to the model. And especially for Alzheimer's disease, this makes total sense, because I've shown you that Alois Alzheimer explored this disease more than 100 years ago, and we don't actually, in the causal treatment, we are not further than he was, but we know much more about the disease. So it's one of the diseases where most literature is available, and no one has an overview about all these 100,000 and more and millions of papers. So we need an approach to access this knowledge. And actually, this is possible because in the last years, the technologies of ontologies and knowledge graphs and other methods of computational semantics allow to directly do this. So the idea was to extract knowledge from so-called knowledge graphs, which are graphs, so organized knowledge structures that contain nodes and edges and directed edges, and can describe the relationship between biological entities, as for example genes, proteins, chemical compounds, and diseases. And one of our partners, the Fraunhofer Institute of Scientific Computing, has developed one knowledge graph for neurodegeneration, so for Alzheimer's disease, Parkinson's disease, and also for epilepsy, which is called NeuroMMSIC. And this contains all this information. And we wanted to make this usable for brain simulation. But to use this, we need a spatial anchoring. So this is one subgraph of this NeuroMMSIC graph. So I hope you can see how it moves. So we zoom now in and see all these nodes. So you see, for example, here there's different proteins and enzymes and these connections between them. And this contains, of course, a lot of information about Alzheimer's disease. But how to implement this into a model when we don't know where these processes take place and where these relationships are located? And to do this, we developed another software, which is called TV-Base, and which I will now shortly show you and explain you, because it will also be part of the talk of next week. So TV-Base is able to map every semantic structure to the brain. So this is now the same graph as I have shown you before, but instead of these little nodes, we have these little brains that are just brain maps that show us where this process takes place, or at least where this semantic term is associated with. And how this is done, we use another software, that contains statistics about the literature corpus. So the literature in PubMed, it's about 32 million papers, contain, of course, a lot of words. And with the help of ontologies, which are organized knowledge structures, we can extract more information about these words. And the idea is that we used an anatomical ontology to extract the stochastic appearance of different anatomical terms in context of different queries to map this query to these brain regions. And I will explain this a bit more in detail, because the idea is in principle very simple, but in the end, it was of course not that simple. So TV-Base relies on the software that I already mentioned, which is Skyview. And Skyview is a search engine very similar to PubMed. It also contains the same papers and the same manuscripts as PubMed. So you can search for something, as for example for Alzheimer's disease or for amyloid beta. And you can also search for controlled vocabularies. So for example, for a mesh term or for a gene term, an HGNC term. And then what happens when you search for that? You get a list of papers. But in these papers have undergone text mining. So the words inside of the papers and the abstracts are marked. For example, in this case, you see the anatomical terms that are part of the Uberon ontology. So for example, cortex, gray matter, precuneus, gyrus, right hemisphere, these are all anatomical terms, and the text mining had marked them as an anatomical term of the Uberon ontology. And they have a number, they have an Uberon ID, and unique ID identifier, so that you can relate this to other anatomical terms. And you can extract statistics about this, because you can count how often a term is mentioned in this paper, and you can compare it to the overall frequency of this term. And the good thing of the Uberon thing is that it's not only giving IDs, it's an ontology, so it also contains a hierarchy. So for example, here you see that the primary visual cortex, it's one Uberon term, and for example, primary visual cortex layer 6A is of course a part of the primary visual cortex. And the primary visual cortex is part of the occipital lobe. And it's also a functional part of the brain and also part of the cerebral cortex. And it's also a Brodmann area. So all these information is part of the ontology. And therefore, it makes a lot of sense to use this ontology to extract the statistics. And what we do then is that we mapped manually in a very detailed process about 1,000 of these anatomical terms to a standardized three-dimensional brain template. So we used the Glasser parcellation, which is a parcellation of the Human Connectome project, and contains 180 regions per hemisphere, and with some subcortical areas, and addressed all of these terms to one or several of these parcels. So we then directly know, for example, the word frontal gyrus, we directly know which parcels in a Glasser template are related to frontal gyrus. And therefore, we also know exactly the voxels on a standard template that are meant by this word. And this allows us to then map, when we have the statistics for the appearance of different anatomical terms, we can directly project it to those voxels, and then we have a three-dimensional map something. And this was the very first interface. Now we also have a GUI and so on. Unfortunately, it's still unpublished, but it will be published soon. And here you can see, for example, we have just looked for memory, and you can then see where it is. And also first validations have shown that it works very well for a lot of different things. So for example, here you see two classic aphasias, so the Wernicke and the Broca aphasia, where we directly see the typical areas, the Wernicke area and the Broca area. And we did this for a lot of different neuropsychological symptoms, but also now for the whole genome and the HTNC ontology, and for almost all mesh terms that are related to the brain. So you can do it with everything. So you can do it with free text, but also with all ontologies that you can imagine, and can therefore map also pathways to the brain, which was the initial idea. And just to show you how, for example, such a map looks like, here we have it, for example, for frontotemporal dementia. We can also export that as a NIFTY, and then combine it together with this knowledge graph to also get the directionalities, and then just have a spatial anchoring of these pathways. And these maps can, of course, be implemented into brain simulation, as we also did it before with PET. So I will also show in the end that this was done with amyloid PET, and we could do it there, but it's also, of course, possible to do it for different receptors or for a particular gene that you are interested in. And everything that you can search for, you can also map it with the software. We also had the possibility to recompute it to almost all atlases. So we have more than 40 atlases where we can put it to. And we also showed that, for example, here just very shortly shown that we could relate it to amyloid PET, again, of the Alzheimer's Disease Neuroimaging Initiative, and could show that the correlation between amyloid PET and symptom maps, for example, memory and language and so on, were able to differentiate between the disease groups, but were also able to predict cognitive scores, so individual cognitive scores. So this just showed that the symptom maps really showed what they should show based on empirical data. And just to complete the vision of how to integrate knowledge to the brain simulation, we can, of course, besides these newly built or newly mapped maps, we can, of course, also extract empirical data, in this case from eBrains. And this is, for example, receptor densities that can be requested by the Zebra client, or also gene expression data, where we probably also will hear about in the other weeks of the summer school a bit. And we also then, because I talked a lot about ontologies, we also built an ontology for the virtual brain that will also be part of one session of the summer school. Therefore, I just want to shortly touch it, that this is an ontology that describes the mathematics of the neural mass models in the virtual brain and the relationship between the parameters, and also the comparability between the models. So it's consists of axioms and classes and so on. And it looks like, for example, this, you can have a look to a particular parameter and see that that is the coefficient of another parameter. It's a parameter of a particular of a function, of a state variable. It's part of a category, like, for example, here, inhibition bound or inhibition. So you can do a lot of things with this ontology. And we also linked it to biological surrogates, so that it can be connected together with the knowledge graphs that are derived from genomics and proteomics that I have shown you before. Okay, and just to show you one use case where we use this for finding new candidates, is that we just mapped a big cohort of entities, actually, from this NeuroMM-SIG ontology that I've mentioned before. which were about 1,000 maps, and then we just extracted the maps with the highest spatial informativity. So the maps that had the strongest patterns, not because they are most reliably. So we also selected the most evident maps, but then from the most evident maps, we looked for those that have the strongest patterns because they are the most interesting ones for brain simulation, but also most interesting ones for a disease like Alzheimer's, where we know that there is a strict spatial pattern. And just by this method, so without measuring any new data, just by the descriptive analysis of the stochastic maps, we were able to figure out very interesting pathways in Alzheimer's disease. For example, here the increased tau deposition was one pathway, glucose metabolism, cholinergic synthesis, calcium binding, epileptogenicity, and apoptosis. And combining several of these maps, we also came to a candidate mechanism of exotoxicity, which is actually exactly the mechanism that I've shown you before that we arbitrarily chose. So there's also one possibility to come to this just by extracting these maps and compare those maps to each other, where we then see how all these factors interact. So for example, here how tau leads through an inflammatory response to the activation of NMDA receptors. And in the end, therefore, to show you that this can really be implemented to a simulation, this is a preliminary work, but we translated the model that I've shown you before with a PET, so I think you know it now well after this talk, and we substituted the individual PET data by TV-based maps of different diseases. So we searched for amyloid beta in context of disease X, and for Alzheimer's disease, we got exactly the same patterns as for PET. So we also saw the slowing phenomena, and that looked very similar. And for the other diseases, so for frontotemporal dementia, healthy controls, Parkinson's disease, and vascular dementia, we did not get this phenomenon. So instead, we still saw the alpha frequencies. So we were also able to reproduce the findings from the individual data with this generic maps, which also shows that you can just directly implement those maps into the simulation. And this is also one of the ideas that we want to focus in our future research. So maybe just to summarize the ideas that I mentioned, we want to improve diagnostics. With traditional machine learning, we already did it with a small cohort. We are currently working on a bigger cohort to reproduce these results with about 1,000 patients. But of course, it's also interesting, especially when we have more subjects, to have a look into deep learning strategies. For the virtual therapy of neurodegeneration, we can combine it with a mechanistic approach to find new targets, especially if you see that we looked into pathways and candidate mechanisms. This is, of course, something that can be brought together with translational research, developing new treatment strategies and new drug targets. So we can really bring also brain simulation to the clinical trials. This is also something we really want to do. But we can, of course, also stimulate brains. This is also then will be part of another week of the summer school. So it can be combined with control theory, but also just with classical brain stimulation, how to bring the virtual brain into different states. And we can explore the mechanisms, of course. I think this was outlined quite extensive also in this talk and in the other talks. And therefore, in the end, we also want to have this TV-Bayes and the TVB ontology combined with the virtual brain, so that, for example, TV-Bayes can provide maps or can provide mechanisms, potential mechanisms. TVBO, the virtual brain ontology, can help to design then a cause and effect model that can then be used in TVB to perform a simulation and to make exactly what we've shown exemplarily with Alzheimer's disease, but then you can also do it for other diseases. And in the end, this should, of course, also lead to to real applications. So as I mentioned, the diagnostics, but we can also imagine that we just directly take data or pathways candidates from this research and translate it really to treatment strategies and try it out, so that we have the whole loop closed, and we have data technology, we have brain simulation, and we have clinical studies all together. And I think this is also what we aim for with translational research. Okay, so with that, I'm now come to an end. I think, sorry that it was kind of a rush. I just shortly show the references of this talk, also for the recording. And afterwards, I would be happy to answer your questions. But before, it's a lot of references. Before, I also want to thank you for your attention, and also thank my colleagues from the